So it's my pleasure to introduce Professor Carolina Lucas. Carolina Lucas is a microbiologist and biologist by training, with a bachelor degree from Universidade Federal do Rio de Janeiro, and with a master's degree in microbiology from the same university, and also a PhD in the same university, correct? After that, she was a postdoctoral researcher at UFRJ and also at Yale University, where she's now as an assistant professor. In her lab, they are interested in identifying the levels of early immunological biomarkers across a spectrum of patients with an immunological profile and clinical manifestations. For this, they use molecular genetics and virology techniques as molecular signatures and as a diagnostic tool to prevent the development of some diseases. So help me to welcome Professor Carolina Lucas.
Okay. So thank you for the introduction. It's a pleasure to be here today. So just this, I think you will start now the immunology classes. So I don't know exactly the background. So our idea, so these classes will be together with Professor Daniel Mucida. Daniel works more with mucosa immune response. And a summary of what I do, I work more with immune response to emergent virus. So I always have worked with virus. I have worked with dengue, Zika, chikungunya, but also SARS-CoV-2, monkeypox, and multiple virus. And these classes will be more focused on the immune response. And I will be more focused on the immune response to viral infections. But Daniel is more classic immunologist. And because of that, we decided to split the class. This first one will be very, very simple, just basic concepts of virus, basic concepts of immunology. And then he will start like the more complicated stuff. So this is the class, if you don't know something, if you want to stop, interrupt, to ask. If it's very basic, we can also skip some slides, so just let me know, because we really thought about doing very basic in the first class. So if you could introduce your name and what is, like, if you are a master, PhD, undergrad, and what is your course, just for me to have also an idea, and maybe the next class I can adjust a little bit based on that. Very basic, just three words, no worries. You can talk more, but it can be just 3 words if you want.
I'll define myself in 3 words. Adenilson, mathematics, biotechnology.
That was great. That is good.
I can try to do the same role. Denise, physics and epidemiology.
Okay.
Cristiana, Applied Mathematics.
Sebastian, Biology, Evolution.
Amanda, Biomolecular Physics and Applied Math.
Tawane, Oceanographer, Megafauna.
João, Ecology, Conservation.
Gislaine, Veterinarian, Infectious Diseases.
Okay.
Mayra, Chemistry and Biochemistry.
Thales, Snake Venom Toxins, Toxoplasma gondii.
Different word.
David, Epidemiology, Malaria.
Erika, Systems Biology and Bioinformatics.
Hi, I'm Ian. I was teaching this morning. I'm a director. Collective behavior and biology, complex systems.
Amanda, Ecologies, Evolution.
Julia, Undergraduate Biology.
Okay.
Same, Nicole, undergrad student Biology.
Mateus, physics and neuroscience, maybe.
Maybe.
Thales, biology, botany.
Cauê, mathematics, Brazilian.
Benjamin, physics, movement ecology.
Ruth, mathematics, epidemiology.
Isabel, animal evolution.
João, biomedicine, immunology.
Julieta, physics, maybe neuroscience as well.
Camila, biomedical engineering, dengue.
Okay. Okay, thank you all. I'm relieved that there's a lot of biologists, immunologists. I was quite afraid of the mathematicians if this will be more complicated, but okay. So this is the outline. So as I mentioned, this will be very basics, and then the second class will be more what I do in the lab, and will be more data. So it's what we did in the last 3 years. So this will be much more data. And Daniel, believe me, it looks more simple, but it's more complicated than that. So this is the time if you have questions, basic concepts. This will be the time. So this is just the outline of this class, so I divided in 2 parts. And again, if it's too long, we can skip some parts. But the idea is to do an introduction to immunology. So I will talk about the immune system, the concepts of immunization, vaccination, and also innate and adaptive immunity, and immunopathology. And the second part will be about the immune response to viral infections. And we will define the property of virus, and again, innate response against virus, and also adaptive response against viral infections, and talk about immune evasion by virus. So the first question is, what are the functions of the immune system? And this will be a conversation, so I want your help on that. So when we talk about immune system, what is the first thing that came to your mind? Defense. So yeah, so if we think, probably the basic thing when we think about the immune system is about defense. But how do you think this defense happens? So what is against, the defense is against what? Pathogens. And this brings something very important. The immune system recognizes pathogens, but how the immune system does not recognize the self. So this is just as important as to recognize pathogen is to not recognize self. And what are the other functions that you can think about the immune system? We mentioned pathogens, but what about, for example, tumors or cancer cells? These are differentiated cells. So this is also another function. And can you think in something else of the immune system? Not only about defense? Regeneration. So this is already like 2 concepts that are very important, and we will discuss across this class. One is resistance, so resistance against, defense against pathogens, but the other is tolerance. So to understand what is self and non-self, and also to induce maintenance of the tissues, and regeneration of the tissues, protections of the tissues. And the third thing that we can think maybe it's about surveillance. Surveillance also comes before all of that. So here we can see like the basic functions. So of course defense, autotolerance, and also immune surveillance. So we have identification and protection against pathogens, but also toxins. And then it comes like the first idea of defense needs to come with the self and non-self, and this theory it is from 1959, and we also have autotolerance, so recognition of own tissues and keeping tolerance, for example, to microbiota. You will see this a lot with Daniel. Daniel works with mucosa and gut, so there's a lot of interaction with microbiota and how we identify and do not destruct our own microbiota. And we also will talk about immunosurveillance, so it's removing old cells, damage and dysfunctional cells, so tissue homeostasis, and also regulation and tissue repair. So these two things I want you to keep like all the time in mind about resistance, but also about tolerance. So when we think about the immune system, all animals, they possess a system of defense against pathogens. And here you can see this is a very old picture, but this shows very well innate immune system. And you can see in red just the adaptive immunity. Let me check if this works. Yes, adaptive immunity. So you can see here, this is evolutionary tree. We can see the evolutionary pathways that have led to the development of innate and adaptive immunity. And these are conserved features from bacteria to higher vertebrates. So all animals are able to defend against pathogens. And here we can see more in details. I think this, I don't know if it's very small. But here we can see more in details, like for example, things that we were going to discuss. So we have PRR, so patterns of recognition, receptors. We have other components of the immune system. We have like lymphocytes, and when this branch of the immune system emerge, then we have other kinds of like T cells. And here was just to show that multicellular organisms and interspecies, there's interactions for cooperation and competition, and this led to evolution of the immune system. So the survival led to evolutionary refinement of the immune system, and we have the emergence of innate and especially of adaptive immune systems later on. So, okay, so if we were about to think what was the first line of defense, we can talk about antimicrobial peptides, so especially defensins. So there's of course more, but defensins could be an example. So these are small peptides that are found, they are very conserved, and they are found among different species. And these are peptides, structure related, and derived from the same ancestral, and they have distinct activities. So they can act against bacteria, fungus, and also virus. So this is the first cellular response that we have, is the phagocytosis. So this was the first mechanisms of cellular response from the immune system. And here you can see images of phagocytosis. And this evolved as a feeding mechanism. And it's also presented in all phyla. So this is a very ancestral mechanism of defense. And with that, this was just to show that we have the innate and the immune system. And as we discussed before, there's the defense, but we also evolved to allow the coexistence with the microbiota to protect defense against infectious agents, and also to initiate repair and remodeling process that restore and maintain tissue homeostasis. So now I will jump, because we will also work with some concepts of vaccines and how to develop vaccines. So what do you think it means, immunization? Yeah, the act of immunization will be, one exposure, and to create protection. And do you know when it starts, the first process of immunization? How long time ago?
By humans?
As an embryo?
Yes, that is our innate protection, but I'm saying like the process of immunization for us to generate protection, so to induce protection.
You mean historically?
Say again?
You mean historically?
Historically.
In China?
In China.
They used the
smallpox.
Yeah, smallpox.
So the first idea of immunization, it started in China a long time ago. And this because there was an observation that people who contract smallpox did not manifest the disease again. People that survived will not get infected again. So becoming infected confers immunity. So this was the first idea of immunization. And this started a long time ago, as he mentioned, in China. And the idea, it's called variolation. So in Portuguese, the word is varíola, but in English is smallpox. And this, what they did usually is to collect the pustules from people that were infected, macerate it, and they used to inhalate. So there was a mucosa vaccine. It was an intranasal vaccine that we don't have it today. But this was in China, as you can see, a long time ago. And the first, of course, the idea of infections comes much before BC, so before Christ. This is in Egypt. So here you can see, for example, this is a person infected that we could, it is a mummification. So they observed that these people also have some scars. So the first description of infection was much before, but the process of immunization started in China and then especially in Turkey. And we will talk about this later on. And then there was a modification. Instead of inhalation, they used this process of variolation. So they collect the pustules and they rubbish this against the skin to generate a kind of immunization. So here, I think this is the, so we have descriptions of immunization, and this started before Jenner. We know that Jenner is the father of immunology and also vaccination, but this process started much before, and late Montagu was a person that was responsible to also spread the idea of immunization. So she was married with an ambassador that lives in Constantinople, that now is Istanbul, and she used to travel there, so she's English, and she used to travel there, and she saw that people over there did this vaccination. So she started writing letters. So when she was a child, and you will see that the variola or the smallpox, it starts long time ago, as I mentioned. It was described in Egypt a long time ago, but it was just eradicated in 1980, in the 80s. So it was a long time that we have a lot of outbreaks of smallpox. And she was infected when a child, and there was also like, there was these scars all across your skin, and people that were not, that usually not died from the infection, they get completely disfigured. So it was something that people real fear about. And she got when she was a child. Her brother also died from smallpox. So when she moved to Turkey and she observed the technique that they use over there, she started writing letters for a lot of people in England to convince people to start the immunization. But of course, we know that a lot of people were resistant to vaccine. We still see that today. And she then did this process in her son and her daughter. And after that, this started gaining traction. And she was responsible for a lot of the vaccination and introduction of vaccination in the West Hemisphere. And this is the first clinical trial that we have. So this was the clinical trial. It's called Royal Experiment. So the Royal Experiment, again, this was before Jenner. And this was also in England. So the Queen and the King, they have a son that was with smallpox. So they decided to do a clinical trial with prisoners that were sentenced to death. Not very ethical, but this was the first clinical trial in history. And this was open, so people could go there and see what was going on, and they could decide if they want also to get vaccinated or not. But this, yeah, the first idea of the clinical trial came from this. But of course, after that, as I mentioned, was not very ethical. But at the same time, this opened and started sanitation and also procedures that help eradication some of diseases. And then this is what we know about the father of immunology, and the beginning of vaccination is with Jenner. And again, Jenner was the first that published the idea and did this in a way that other people could also repeat. So that's why it was considered the first described the immunization. And differently from what I explained so far, what Jenner did was to use cowpox. So cowpox is not, it's not smallpox, it's not vaccinia. So cowpox infects cows, and what he observed that people that handle cows would also get some scars in the hands, but not a very infectious disease, or usually will not progress to a worse disease. So he used the cowpox to inoculate and vaccinated other individuals, but of course at the time he had no idea what was the pathogen. We couldn't isolate the pathogen, and it took 2 centuries to eradicate smallpox. And with Jenner, it also starts the anti-vax movement. So we all know about the idea of changing as a crocodile. So this is long time ago already exists. They all think they will turn into pigs after receiving the cowpox vaccination. So these ideas are not new, unfortunately. But so this is a painting showing that people were afraid because the cowpox was derived from cows to turn into a cow. So as I mentioned, at this time, we don't have the idea or what was the agent causing the disease. And because of that, it was much more difficult to develop a vaccine. So this idea of disease causation came with Koch. And of course, this will not work for virus. But this idea, this works very well for bacteria, but this changed the culture of microorganisms, and this also changed the generation of vaccines and therapeutics. So the Koch postulates, it says there's 4. So the first one, the microorganism must be found in animals or people that are infected, in all of them, and not in healthy animals. So if you try to culture there, you should see something different that you cannot find in health animals. The second postulate, it says that you should be able to do a pure culture. So as I mentioned, this will work very well for bacteria, but this doesn't work for virus because virus need cellular to replicate, to grow. And then the third postulate, it says that if you isolate and inoculate in the animal should die again, and then you should again be able to isolate this pathogen. So with that, we start seeing a bunch of development of vaccines. So the other vaccines that were developed a few years ago after the Koch postulates was the vaccine to chicken cholera, and also anthrax vaccines to humans, and rabies vaccines. This was Pasteur. And here you can see what he did was to use a very aged bacteria colony culture. So this was a kind of attenuate. They didn't know exactly how to attenuate, but it was attenuate culture. And he injected this in a normal chicken, and this chicken wouldn't die. And if you inject a fresh bacteria in the chicken that was immunized, the chicken wouldn't die, but a chicken that was not immunized would die. So this was one of the first vaccines described. And as I mentioned before, then he also did develop vaccines for humans and rabies vaccines. This was also very close when we developed the first BCG vaccine that is still used today. Then here so with these discoveries, we are able to isolate microorganisms. We are also able to develop vaccines and also to better understand the immune response. So at the same time, after these experiments, we have 2 researchers that discovered the antitoxin that we know today as antibodies. So the antibodies, they observed that the animals that were immunized with these attenuate pathogens, they collect the serum, and they discovered that in the serum, if you just transfer the serum from another animal and then do a challenge, try to infect, they would be protected. So the idea of something in the serum was protecting started here in 1892. And this is the idea, of course, of also passive immunity. And here is also raised the concept of an antigen. So here is when we start working with antibodies. So as I mentioned, here we already, the microphone. We already have an idea of immunization, active immunization and passive immunization. So the active immunization is when you use the direct antigen or microbial and you do a challenge, and this will induce, the major difference is this will induce memory. But if you just transfer the serum or just transfer the antibodies, this will be able to protect you depending on the time, but this will not induce a memory response because we are only transferring antibodies but not memory cells. So much, if you remember, like here it was, the idea of protection came of antibodies in 1892, and the protection through T cells, it just happened much later in 1987, when we have an experiment of that we just transfer T cells, and this is again, this is protection against a bacteria, listeria, and animals that we have the transfer of T cells, they were protected compared to animals that did not receive T cells from an immunized animal. So the idea of T cells protection just came much later. So with that, we will start talking more specifically about innate immune response. This was just more about history, and adaptive immune response. So this looks a little bit complicated, but this is just a general sense of the response, the immune response. So the innate immunity, we will talk about the cells, like dendritic cells, macrophage, NKs, but also we talk about defensins. We will talk about cytokines like interferon, and also tissue damage, tissue adaptation. And in the adaptive immunity, we can see that there are some cells that do this crosstalk between innate and adaptive immunity. And we will talk about like T cells, B cells, and antibodies. So here, I just want to give you the major characteristics of each innate and adaptive. So if we talk about sensing, how sensing the pathogens, how the immune system can recognize pathogens, and this will be very different from the innate and the adaptive immunity. So the innate immunity, the cells from innate immunity will codify PRRs, so our germline encode pattern recognition receptors, and here we can already see some of these receptors, but we could separate these receptors. Like, for example, these receptors could be cellular receptors or humoral receptors. So here we have an example of the TLRs, toll-like receptors, are in the cells, but we also have receptors that are soluble. So these are in the plasma, for example, like complement receptors that are in the plasma. Then in the cellular receptors, we have intracellular or cell surface. So we have like TLRs in the cell surface or dectins in the cell surface. But we also have intracellular, and we will understand why. And we can also separate receptors that are sensors and different kinds of receptors that are more phagocytic. And one important thing, so the innate immune response, they have a limited repertoire. So they can just identify some structures. And these structures, of course, will be common for pathogens and non-common for our system. So it's not common in the self molecules. So we'll have some patterns that will be able to be identified by these receptors. And they have multiple receptors, but there's a limitation of what can be recognized. When we talk about the adaptive, okay, go ahead.
I have this question that is more related to the evolution of the immune system itself, because when we think about some receptors like the TLR receptor, we have this big chain that is conserved across all the TLRs, but then we have the recognition part that vary across the different TLRs for them to be able to recognize different patterns. And I have this question about how this repertoire were formed, because thinking that we have the TLRs across different animals and for a very long time. So why we are not like accumulating mutations in this region that can vary across different TLRs and being able to recognize more types of patterns? Do we have some type of work?
Yeah, that's a very difficult question. This, of course, is a very important question, but there's a limitation. This we will discuss more by the end of the class. There, of course, there's a need, the immune system co-evolves with pathogens. And you imagine if pathogens keep evolving, we should also keep evolving. But there's a limitation of what can be done by innate receptors. Of course, we evolved to recognize, and we'll see more clear, specific patterns that will be shared across a bunch of, but there's a limitation, and that's exactly why that limitation was suppressed with adaptive immunity. So there are, maybe, I don't know, maybe people from evolution can also participate in the discussion, but there's of course a limitation of what can be done, and what we know is that adaptive evolves exactly to try to suppress this kind of things that are limit for the innate response. I think in the end, if they're still not clear, I think I can go further, but that's exactly like the adaptive immunity evolved to overcome the innate obstacles.
Yeah, no, I got it, but the question is exactly where this limitation comes from, because we have, like in mice, we have more TLRs than we have, and then they also have adaptive immunity, so why this specific condition in us, in humans? But
why there's a limitation of some kinds. We have different kinds. I don't know if the professor wants to add something on this. Excuse me. I think there's a limitation, and of course, how the adaptive evolved to try to control. One thing that we, again, I probably will not answer very directly your question, but to be infected, to have a disease is a rare event. And definitely it was very successful the way that we evolve. I don't know exactly like why some kinds of TRRs, we can imagine that maybe some animals that have lower structures, for example, don't have the recombination of T cells, will need to have an addition of a bigger repertoire. But I will not try to get too much into that because this is definitely not my area. So, as I was mentioned, the adaptive immunity evolved to overcome the innate obstacles. And here we can see that the B cell receptor and T cell receptor, they are evolutionarily related. And we have this region that is a variable region, a constant region, and a transmembrane. And these cells will be more, more diverse, and they have an unlimited repertoire, and we will understand why. And these cells are in the cell surface. Of course, the antibodies can be secreted, but the T cell receptors will be always at the cell surface. So the difference of the innate and adaptive, so the receptors will have a higher specificity and higher diversity. So here again, we have the concept of antigen. So the antigen is a molecule or a molecular structure that is recognized by the TCRs or BCRs, as I showed before, the TCR from the T cell and the BCR from the B cell. And an antigen can have multiple epitopes and can be recognized by different TCRs and different BCRs. And each lymphocyte has a single antigen receptor. We also learn about this idea of clonal expansion, and the human repertoire has 10 to the 7, 10 to the 8 of lymphocyte receptors, and the antigenic receptors are generated by somatic recombination of a limited number of gene segments. This will get more clear. So as I mentioned, the expression of antigen receptors is clonal. So what does that mean is that after an antigen recognition, to activate the T cells, as I mentioned, there are some cells of innate that do this crosstalk with the adaptive. So these are basically APCs, or cells that recognize antigens and present it to T cells. But after recognition of a foreign antigen by a T cell, you can see that there's only expansion of this type of cell. And these, of course, raise some questions. There's one question, is there's only one cell that is specific for that antigen, how they encounter. So this, of course, needs to be localized. So that's why we have different kinds of structures to to increase the opportunity of dendritic cells to present it to T cells and to encounter these specific cells, to then generate this expansion. And the other conceptual problem is that if the generation of the repertoire is random, it was expected to us to have a number of lymphocytes that also recognize self-antigens, and that doesn't happen. So this also, this is the idea of clonal deletion. So this is the first tolerance mechanism. So the first step is to delete all clones that are able to recognize to auto-antigens. So this happens in early life, and this is the first tolerance mechanism. And as I mentioned, this, the diversity of the antigenic receptors is unlimited. So this happens by somatic recombination. So we receive the genes from our parents, and there's this recombination. And as a consequence, we have a limited number of genes, generate a large number of different proteins. Each cell will have a unique specificity, and the rearrangement involves an irreversible change in cellular DNA, and all the progeny of the cell will encode the same receptor. Is that clear? Okay. So this was the basic concepts of innate and adaptive immune response. So innate adaptive immune response will confer memory because it's specific for an antigen, and this will generate a specific protection. Specific recognition through antigenic receptors, or receptors that can recognize antigens, and it's diverse, so it's generated by somatic recombination. There's clonal expansion, so each lymphocyte only expresses one antigenic receptor, and there's activation of lymphocytes with the same specific as the original cell, the same clone. So what are the mechanisms? So this is the basic part of the immunology that I mentioned. So with that, we can jump for what are the basic mechanisms for pathogen clearance. We'll talk more about this in the end of the class, but this is usually it's a very typical, if you are from immunology course, this will be a question in your exam. Just explain, you see a bacteria, how you will deal with that. And there's a huge response about that. So, but try to be like what we learn. So this is the more simple this can get. So we have intrinsic defenses. As I mentioned before, disease is a very rare state. Consider how often we are exposed to pathogens. So we have intrinsic defense, then we have innate immune activation, and then if even the pathogens can go further from the innate response, we will have the adaptive immunity. And here was just showing more in detail. So imagine we have a pathogen. So we have a bunch of structures like skin, mucus. If the pathogen is able to cross that, then we have recognition by innate cells. If the innate cells are not able, so we have recruitment of effector cells. Usually this is in 4 hours to 96 hours. Then we have the transport of the antigen for lymphoid organs, and then we have the activation of innate immune response. Usually it happens in the day 4 of infection. And then usually this will be, there will be a clearance by B and T cells, and also the generation of memory. Again, this is just very simple. We'll talk about some steps, and then we will discuss more about specific pathogens. And one thing that I want also to be very clear is, of course, when we have the pathogen clearance, we also need to have a return to homeostasis. So we have a phase that we call contraction. So after expansion of the immune response, like after expansion of T cells, after production of antibodies, we need a phase of contraction to minimize damage, and also to minimize inflammation, and to tissue homeostasis. And the majority of the cells will die by apoptosis. This is a control of the immune response. And we will only have the memory cells that will be important in a second reinfection. So the second part, again, will be also very basic, and this is about virus. Super basic about virus structure. definition of virus, the property of the virus, and then we'll talk more specifically about adaptive and now that we saw what is adaptive immune response, innate immune response, how that will go with virus infections. So, okay, let's start again. How long virus has been around us? Of course, we don't know that, but it was a long time ago. And we mentioned that viruses exist everywhere life is found. In virus, of course, we usually always see virus as something negative, but viruses are essential to life, and they have an impact in ecology, in evolution, and in all organisms from bacteria to humans. So there are some, I'm not going very much on this, but there are some ideas of how the virus, what is the origin of a virus. There's 3 classical hypotheses. So the first is the virus first hypothesis that states that the virus were co-evolved with the current cellular hosts. So we have genomic material like DNA and RNA, and then there's this idea that the virus came first, and then we have the first cell, the early cell, and then the modern cell. Then we have other ideas like the reduction idea or regressive hypothesis that states that viruses are remnants of cellular organisms. And then we have the escape idea or progressive one that states virus arose from genetic elements. So if we think on some virus like retrovirus that are able to integrate to the genome, maybe this idea will be better. Thinking on a virus that can integrate, and then you imagine, oh, it was integrate, and after that was able to jump and to generate a pathogen. And then we have the second idea, like that we think maybe in DNA virus, that this DNA virus, they just at some point separate from the cell. Again, I mentioned I will not go very much onto these ideas, but this is just some hypothesis, what will be the origins of some of the virus. And as I mentioned before, we talked about smallpox before. Definitely one property of the virus is that virus can be transmitted for many years, and they can be maintained in the population, in populations. As we talked before, like the first time that was described smallpox in Egypt was probably 3,000 years BC, and the last case, it was in 1878, and this was the first described case of smallpox. So it was maintained in the human population for very long. So this case was in Africa, and the eradication happened especially in the 66 to the 80s. And as I mentioned, not all viruses are pathogenic. Some viruses are beneficial and has beneficial contributions. So viruses are able to infect organisms in all domains. So they can infect bacteria, they can infect vertebrates, and they are the main agents of evolution. So viruses are able to induce this horizontal gene transfer, and they are also very important to the ecosystems. So again, this is not something that we will talk today, but definitely, if there's a disappearance of virus, this would impact all kind of life. And for example, in oceans, we know that viruses are the major predators of bacteria. This will completely change. And we also know that we are viral. 80% of our genome, it's composed by endogenous heterovirus. And this was left behind as a result of infections that humanity and primate ancestors suffered millions of years ago. And of course, this was an example of how can we use virus. So we developed multiple research tools like biology and disease evolve because like we have new vaccines, we have new drugs, we have new therapeutics, and we will discuss a little bit more about this. Like for example, many of you work with molecular biology, and we have RNA transcriptase that was just discovery after we understand more retrovirus, and there are many examples that could be used. But as we talked before, the majority of viruses have little or no impact in our lives. However, there are some viruses that are capable of decimating large human populations. This is exactly what I work. So here you can see the major pandemics that we have. Of course, not everything that is here is from virus. We have a lot of bacteria, we have the plague, we have also cholera pandemic, but we have a bunch of viruses like Spanish flu, swine flu, COVID, Ebola, HIV. So here, can you identify what are these viruses? Like, for example, here, what will be this virus? Smallpox. This is more complicated, but yeah, it could be a virus, but it's yellow fever. Here, it's Ebola. There's recent outbreaks of Ebola. We also have very good vaccination, and because of that, we have polio. And here, it could be a few years ago, but it's not. This is Spanish flu. So this is just a journal from Rio. This was in 1849. And this was already, of course, before we weren't expecting to have a pandemic a few years ago. But this before was epidemic in Rio de Janeiro. This was yellow fever. And here was, again, this was a magazine. There was some criticism about the government. So this was the yellow fever talking with the government at the time because they were collecting around 100 to 200 cases a day of dead people. So the yellow fever at this time in Rio caused 1/3 of people get infected, 1/3 of the population. There was a lot of death, and this was one of the examples of they call the carnival of this time. So I know that today this is not very distant from what we passed, but if it was like in 2019, this would be much more distant. This could be happening today. We could have some comparison of some charges today that we also have a lot of discussion and government issues on dealing with a pandemic. So yeah, this is just to show how some viruses were introduced to other regions. This we call dance of that. This is also yellow fever. So the yellow fever, it was known in Africa. It was identified in Africa, and yellow fever was imported, the virus and also the mosquito, with slave ships from West Africa to other regions. And the first outbreaks, there was outbreaks in the US, there was also outbreaks, but this is just to show you how the yellow fever also could spread at the time. And of course today that's changed a lot with the dynamics, but this was how we have some, of course the mosquito, it's not very well established in some regions. This is changing now. So we have mosquitoes much better established. Even in Europe, we are having more cases like, for example, chikungunya that we never had before because the mosquito is adapting. But this is just to show some dynamics and how that happens in the past. And this is also the first eradication campaigns. So yellow fever, the first case of yellow fever in Central America and South America was in Mexico and Panama. And they started, Cuba and Panama started the first eradication campaigns that was then imported to Brazil and was used in Brazil. So this was some examples of what happened at the time. So here we have the fumigator that is still used in Brazil today. Here was how they changed the sanitation and health programs to try to change that. So this was imported from Cuba, and this was imported from Panama. And this is in Rio, and here how we started the first eradications campaign in the country with Osvaldo Cruz and Emilio Ribas. And as I mentioned before, the cases from yellow fever, it took like 50 to 60 years to start eradicating. And we still know that it was, we still have re-emergency of yellow fever, even in yellow fever, and emergency of a lot of other pathogens. So, and yeah, this was the quarantine chamber in the past. So if we were complaining about staying at home, it was worse in the past. So there was this quarantine chamber was used for 3 diseases: yellow fever, cholera, and plague. And usually people will stay here to 1 month to 3 months. So okay, with that, we start the virus, like starting the definition of the virus, how is the viral particle to understand, how is the immune response to the virus. So the virus definition, it started that the virus was an agent that could be passed through filters. So that was the first idea. And this was a new group of a pathogen, because they use filters that could not, any kind of bacteria could pass, but some viruses were able to pass, so this was identification of a new group. And then it came the idea that a virus was a simple microorganism. Then the virus was a type of nucleic acid. And then viruses were considered organisms again. And then we have the first definition. Viruses are not considered organisms. Here they were considered, like in 1957, they are considered biomolecules. So they are strictly intracellular, and they have potential to be pathogenic with an infectious phase. And they have one type of nucleic acid, and they are not able to multiply outside of a host cell. So this idea finally disrupts the idea of the virus as an organism. So it's dependent of a host cell, and we already know that it has nucleic acid. So this is the definition. So we call the virus, the name came from contagion, Latin, vivian fluid, because contagion vivian fluid, again, this was able to pass through a filter, and this means slim liquid or poison. So this is how the name, this is the nomenclature for the virus, and this is an infectious agent. It's a small size that could not be seen in the light microscope, and it typically consists of a nucleic acid and a protein coat, and it's not able to multiply in outside of host cells. So it's wrong to say that virus reproduce, it's wrong to say that virus die, because it's not an organism, so it grows, it replicates. And again, so the virology was born from botanic. So as I mentioned, the first description of a virus came from the tobacco virus and mosaic tobacco virus. And this was reported in 1892. So, and again, this was a filtration. The virus were able to pass. So here we have a healthy plant or a sick plant. And they were able to see that the agents that caused the disease was able to pass through to filters. And this was confirmed later on by another researcher that gave the name. And then we have the first, it was born virology. So the first animal, virus described was the foot and mouth disease. Do you guys know what is foot and mouth? What is the name in Portuguese? So, febre aftosa. Okay. So this was also, again, this was an agent of this disease, was filterable, and was the first animal that was identified. Then we have the first human virus identified was the yellow fever, and was identified by an American. And they also confirmed at the time that the yellow fever was not transmitted by direct contact. They also confirmed this was a theory of a Cuban doctor that was transmitted by mosquitoes. So how they did that? They just get a mosquito, the mosquito bites someone, they collect the mosquito, and they transfer, and the mosquito bite another person. And they also identified that this should happen in 3 days. If this shouldn't happen in 3 days, you don't get infected anymore. So this was the first experiment showing transmission by mosquitoes. And this was the first time that the viral structure was identified in 1935. So this was a chemistry, again, the same virus that was identified before. And after crystallization, he concluded that viruses were also formed by proteins. And this was also the beginning of molecular biology. And okay, so the viral particle, a virus is very simple and is an agent that is just formed by nucleic acid and protein. Of course, viruses can have more, they can have, for example, lipids or can have other structures, but the basic structure of a virus is genetic material and a protein coat. And we have two kinds of virus. We have enveloped virus and non-enveloped virus. So what do you think will be more resistant, an enveloped or a non-enveloped virus? Enveloped? Non-enveloped? Okay, so non-enveloped viruses are much more resistant. So usually because the envelope is a lipid, and the envelope, it's always derived from the host cell. So if you imagine a lipid can be treated with a detergent, can be treated with ethanol, it can be treated with multiple reagents, and the non-enveloped usually are more resistant. So usually virus, for example, if we think enteric virus that needs to go through the stomach, for example, they need to be much more resistant. So imagine an enteric virus will be enveloped or non-enveloped? Non-enveloped. So usually we can already have an idea, depending on how the viral will generate infection, we can have already idea of the structure. So as I mentioned, the viral particle has a genome. The bigger the genome, the bigger the complexity. And the genome can be multiple things, DNA, RNA, and multiple combinations. And the proteins can be, we call structural proteins and non-structural proteins. So here we can see the envelope, we can see the capsid, the membrane, and the genome inside. And here usually when we see the genome of the virus, so here are the different genes, it's very easy to understand. So if we have this kind of, with the names, if we have the name C, E, usually E is for a gene that will codify an envelope protein, a C capsid protein. If it has an S, it's non-structural. Non-structural protein 1, 2, 3. So this is like the nomenclature for virus. And usually the structural proteins are related to genome protection, cell binding, and envelope cell membrane fusion. And the non-structural proteins will be related, these are the viral enzymes, will be related with replication, are transcription factors, and also are important to interaction with the hosts, a lot of times to interfere with the host immune response. So here's just some nomenclature of virus. So when we talk about a virion, is a viral infectious particle. So it should be complete and should be able to infect. Some virus, for example, HIV, the GAG protein itself can generate the structure of the virus and can get out of the cell. But there's no genome, there's no nothing. So that's not a virion because it's not infectious. Will induce an immune response? It will, but it's not infection. So that's not a virion. The viral envelope, as I mentioned, is usually a bilipidic layer. It's always derived from the cell and could be derived from the cell membrane, but could also be derived from other membranes like ER, endoplasmatic reticulum, or any other membrane intracellular. The capsid is a protein shell that protects the virus, the genetic material, and usually consists of repeating structures, and it's made of proteins. So inside, usually we can see the capsid, and the main function is to protect the genome and transfer the genome from one cell to another. And the nucleocapsid will be a mix of capsid and nucleic acid. And here we can see, again, some viruses that are enveloped, some that are non-enveloped, and we can also see the sizes. But here we can see more diverse structure of the virus. So there are kind of structures, all kind of size, but of course viruses are very small. So we have the smallpox is a very big virus, herpes virus is a big virus, and we have very small ones like the parvovirus, for example. And the viral genome, it's very important when we think about immune response and we think about sensing. So DNA and RNA are molecules that we have in our cells. So something needs to be different, or it needs to be, to us to generate an immune response against that, something needs to be different. So what can be different? The structure can be different, the location can be different. So this here shows what is the host genome. So we have double-strand DNA. We also have mRNA molecules. We also have ribosomal RNA, transfer RNA, small nuclear RNA, or non-codifying RNA, and these all have different structures. And then when we think about virus, these structures will be different. So virus can be double-strand RNA, single-strand RNA, circular, linear. They can have structures that looks very similar to mRNA, but can also have single-strand RNA that looks very different, double-strand RNA, or with different kind of modifications, and also different kind of, the products can also be very different. So for example, do we have DNA in the cytoplasm? Well, the DNA is only in the nucleus. So for example, if a virus that is an RNA virus, SARS-CoV-2 is an RNA virus, dengue is an RNA virus, there's multiple RNA virus, and they are positive virus. So the RNA positive virus, they look very much like an mRNA. So if they are in the cytoplasm, they probably will not be identified, but they need to replicate, and they need to do a double-strand RNA. We have double-strand RNA in the cytoplasm. So we have some, but they look different, but it's not common. So if they are in the different region, they can also be sensed, and we will have molecules to sense if something is wrong in the different location. For example, so in the case of an RNA virus, one strain of RNA, it needs to do another strain of RNA. What is our polymerase? We do DNA from DNA, right? So we have a DNA polymerase dependent of DNA. We can also do RNA from DNA. So is an RNA polymerase dependent of DNA? Do we have an RNA polymerase dependent of RNA? We don't have that. So viruses that have that need to codify this kind of molecule, they need to have this kind of polymerase. This could be a target, and this could also be sensed by the immune response. So the virus classification, we have different kinds of virus, and it's very complicated, the classification. It depends. You can classify by the nucleic acid. It could be DNA, it could be RNA. You could classify by the symmetry of the capsid. You could classify by the host. So there's different kind of classifications. And this, for example, is a classification that shows by nucleic acid, by the symmetry of the capsid. And again, it's just to show you that it's complicated, and usually it's not just one type of classification. And one typical classification is also by replication. So we have all virus needs to do mRNA. So we can go from DNA virus that will need to do mRNA. They probably can use the cell machinery because we also do that. There are other kind of viruses like double-strand RNA, single-strand RNA, that they need to use their own machinery, at least one polymerase or one enzyme that is necessary. Let me check the time. I think, oh, go ahead.
Just a technical question. What is the difference of the positive single-strand RNA and the negative?
Yes, so the positive single-strand RNA can be identified by our, can be directly translated into a protein. This is a positive sense RNA. The negative sense RNA, usually it will be the genome of the virus. The genome of the virus, so it's a complementary sense of the positive RNA is the negative sense RNA. So during the replication, if a virus has a positive sense, it can be directly translated into proteins because it looks like a lot like our mRNA. But to replicate, you will need to do the negative sense to then generate the positive sense again. This is the replication itself that will be encapsulated to generate new virus. And again, the negative sense can be used in a lot of ideas. So for example, when we work with virus and we want to measure RNA, we all did here a bunch of PCRs. Usually PCRs, they only measure, they can measure, for example, DNA and then convert to RNA to see if there's the viral genome there. Is that a measurement of viral replication? No, because mRNA is not a measurement of viral replication. So to measure replication, we need to measure the negative sense strain, or we need to do other kind of assays like plaque assays that we know the virus is infectious. So we can also use different kinds of detections to understand. But a simple answer is just is a negative sense from the positive RNA. It's the negative.
And they usually differs from species. So one species is single negative, or
different strains. Yeah, different. Like for example, the positive strain RNAs. We have dengue, we have chikungunya, we have SARS, and the negative strain usually are the kind of emergent virus, the dangerous ones that we talk about. A lot of them are negative sense. The ones that are emerging hemorrhagic fevers ones, Ebola, they are different.
And for example, for the PCR, it's easier to detect one over the other. How do you?
Yeah, you just need to do the complement sense RNA.
Yes.
So again, you will need to use an RNA polymerase, RNA dependent, that the virus have. So you can generate RNA from RNA, and we can then do the negative sense.
Okay.
So here I think I will, okay, go ahead.
But how does because the microRNA, when they first got out from the nucleus, they are double strand, right? How they are not recognized by
Say again,
the microRNA.
The microRNA,
When
the mRNA, when they came out from the nucleus, yes.
So how they are not recognized by immune?
They are different. So the mRNA is not recognized. I will talk more about the sensing that, the sensors that can recognize that, but definitely if it's just exactly the same, you see this is our mRNA, a poly A, and the structure, if the virus has exactly the same structure, this will not be sensed.
No, yeah, I'm not talking about the messenger. I'm talking about the microRNA, the one that will, they use to RNA interference.
Yeah.
So they have the same structure?
There's different sensors. They will not be recognized. Usually very short are not recognized. Okay. Long are, and with some modifications are. But again, it's usually we evolved to not recognize the self, and we usually do not recognize very small.
Mm-hmm.
Yeah, here it was just to show you like what are the contributions of virology, but maybe I will skip or go like very fast on this. But this was a lot Nobel Prizes over the time. So we can see here, this was the first person propagate yellow fever in the chick embryos. That this was, we could successfully produce the attenuate vaccine. And the vaccine was safe and it's still used today. This was a Nobel Prize, and of course this is also like
So the attenuation here, is it just waiting for random events to occur and therefore to have a virus that just happens to be attenuated, or was it directed? Did he have some means of attenuating the virus?
The initial attenuation was, for example, this attenuation was from multiple passages in eggs. So after some time, if you do multiple passages in eggs, the virus adapts more to the chicken cells than to our cells. This is the kind of attenuation, and usually it's multiple passages. Today, of course, we have other kind of attenuations. We have chemical attenuations. We have all kind of reagents that could be used, like the vaccine. The COVID vaccine is an attenuation by formaldehyde. Yeah, but
cool. Thanks very much.
But in the past was this way, and we still do that. So, like for example, today we are working with monkeypox in the lab, and we have the monkeypox. The smallpox vaccine, they use vaccinia, and we are using vaccinia, but the vaccinia grows so well in chicken cells, we want to compare the response to vaccinia to monkeypox to see what is the difference, but the vaccinia grows so well in chicken cells that doesn't grow in any kind of mammal cells. So we don't have plaque assays. We cannot do plaque assays because it doesn't infect very well. We can do in chicken cells, but the chicken cells grow so slow, and no one thinks about growing eggs today. So, and And because of that, we are having multiple problems. Also because we know COVID, there was a lot of talk about being a gain of function and being doing in the lab. So because of that, another way that we could do is to use a GFP virus. But at this point, we cannot work with GFP monkeypox because that could be a gain of function. So we are very, very restricted. So we are almost going back to X. But yeah, this was the kind of attenuation. Microphone.
I have two questions. The first one, do you have attenuated COVID vaccines? No.
Yes, this is the vaccine that was very used here, is the one from China and Bhutan.
Coronavac is attenuated or?
The Coronavac is inactivated.
Inactivated.
Yeah, I'm sorry, it's inactivated.
But
it's dead.
Okay.
It's completely dead. Yeah, we have attenuated. It's like this, the monkeypox and the one that was used in Brazil, it was attenuated, but it was from vaccine. It was a different virus. And we still use it. I don't know if you have now the Genius vaccine being used here, but the previous one was Vaccinia Ankara. It was attenuated.
And what was the last thing you said about the
Genius?
The virus?
The virus itself? Yeah, we use Vaccinia to do the Vaccinia. So the there's 5 orthopox viruses that are known to infect humans. We have smallpox, we have vaccinia, we have cowpox, we have monkeypox, and I'm missing one. Cow, monkey, vaccinia, small. Okay, I will remember at some point. Almost all of them are zoonotic virus. They infect humans and animals. Only smallpox is only humans. All the others are zoonotic ones. And we are not using a very specific vaccine for monkeypox. We are still using the vaccine that we use for smallpox. And that one was done with vaccinia. Like Jenner did with cowpox, we then did with vaccinia. Oh, it was the cowpox, the 5.
The question probably comes because I am very new to this, but I tend to understand the difference. Well, I think I understand the difference between inactivated and attenuated in the case of a bacteria, for example. But in the case of a virus, I have difficulty in understanding what would be a virus that was killed and a virus that was simply attenuated. Because
so again, if we used, I don't know, if you use a chemical reagent, that will definitely kill the virus. Sometimes we can use a chemical reagent that will just change the antigenic part of the virus, for example. But the difference, like attenuation process, it could be, we do kinds of attenuations, like I mentioned, in the eggs. You just pass the virus so many times in eggs that he will get more, he will start to change, and he will enter more easily in an egg cell than in a human cell. So he can replicate, but not very efficiently. The other way of doing, we also do, for example, we passed virus among mouse, a lot of mice, and then we use that is adapted to mice. We call that adapted to mice. That is a way of attenuated. We still have some vaccines. So for example, we have the Sabin and we have the Salk vaccine. The Sabin one, this is vaccines for polio. One is attenuated and the other is inactivated. And we use together one, because usually inactivated vaccines usually don't induce a very good immune response because they are dead, the virus don't replicate it. So usually we need to use an adjuvant, as we have today for the COVID vaccine. Is inactivated but has an adjuvant that will boost the immune system. Inactivated, definitely they work much, attenuated definitely work much better because there's still some replication going on. So you can induce better the immune response and you can generate it better the memory. But at the same time, they are more dangerous. And especially today that now we have a lot of people like pregnant, immunocompromised, you will not want to use this kind of vaccines. So of course, we evolved to more safe ones like mRNAs, there is just subunities vaccines like hepatitis. But attenuation, it's still alive. It's just not adapted for that host. And inactivated is dead.
The amount of pathogen that the body will have contact with will be the exact amount you input in the system, whilst in the case of an attenuated one, it will replicate a bit.
Yes.
Still.
Yes. Yeah. So there's also like the virus. This was the first time that was demonstrated that influenza could agglutinate red cells. This is a very old-fashioned way of doing rapid tests. It's how it evolved. So it was again using different kinds of virus and measuring and quantify virus. Then we have the demonstration of the bacteriophages mutate, and this was helping. This was also a Nobel Prize, and this helped to understand the idea of antigenic variation. We will talk about antigenic variation later, but we have a lot with COVID. We saw a lot of antigenic variation when the virus is able to mutate to escape the immune system. We have different kinds of antigenic variation. We have shifts and drifts, but we'll talk about this later. This was the first time that the poliovirus was grown in tissues. So this changed a lot how we understand virology, because now we could isolate. Again, I show you the postulates. This was so early on, and they could already isolate a bacteria. To work with a virus to isolate, it happens much later. Then we also have the discovery that this kind of virus was from DNA. This was also a Nobel Prize and the understanding of the DNA. We have the discovery of interferon. We will see a lot what is interferon. And yeah, we'll talk about cytokines. So when we talk about first line of defenses to virus, we will talk about a lot of sensing. We'll talk about interferon, and we'll talk about CD8 T cells. These are the major cells for viral infections. And this was discovery of interferon. And here we have the discovery of hepatitis B, also the development of the first vaccine against hepatitis B. It was also a Nobel Prize. Then we have the discovery of the reverse transcriptase. This was a huge jump in molecular biology. Finally, we were able to understand this was by the study of retrovirus. So now we could use enzymes that help us to develop different kinds of molecular techniques, and also to understand the pathway of genetic information from RNA to DNA. So this is just to show all studying virus what were jumps that we could give in different kinds of areas. The antigen recognition by cellular immune system was also a Nobel Prize. This was related to MHC and how cellular immune response were generated. I'm sorry, I'm just going fast because I think this is just for you to have an idea. And here, I'm stopping here, but definitely we have much more discoveries related to virus. But this was the identification of HIV in 1983. And we could talk about Charles Rice that just the Nobel Prize received the Nobel Prize a few years ago, and about hepatitis C and the treatment with interferon that helped to the treatment of so many people. And we could also talk about the vaccines for COVID that were generated in just 1 year post-infection and saved multiple lives, and now we are back to life again. And of course, viruses are also related to cinema, are also related to human creativity. So when we think with werewolf, I think it's wrong, I'm sorry, werewolf, this was described after seeing people that have rabies. The first description of werewolves came from that. And the description of zombie it's supposed to be born of a mix of a person with rabies and influenza. So, okay, so this is the last part. And again, if we don't finish, I can start I think you already know very basic concepts of immunology. You already know very basic concepts of virus. And even if I don't finish today, we can talk this prior to my other class. and this will be much more focused on research and just about data. But what are the basic protection against virus? So, as I mentioned before, I really want you to understand the idea of resistance and tolerance. This was proposed by Ruslan Medzitov and also Miguel Soares. And so we have resistance mechanisms are related to pathogen control. So mechanisms that are related to virus that are resistance mechanisms. But usually the resistant mechanisms were the focus of the research for many years. But the capacity of the host to adapt to the burden caused by the pathogen, so to adapt by directly damaged by the pathogen, but also directly damaged by the immune system, these are called disease tolerance. So here we can see that the pathogen can cause direct damage. So a virus can induce cell killing, cell death. And at the same time, the immune response also can cause damage. So we saw this very well during the pandemic. So we knew that people that did not control the virus very early on, they also generate a very robust inflammatory response, and the inflammatory response was responsible for the disease itself. So the ability of the immune system to control the damage that is caused by the pathogen, but also that it's caused by the immune system, this is called tolerance. And we can think, for example, the example of SARS-CoV-2. We think that children have very high resistance and very high tolerance. So they have, for example, very good interferon response in the mucosa that control the virus early on. This is a resistance mechanism. But at the same time, they don't have the induction of a lot of macrophages in the lungs and inflammatory response. And this is related to a lot of tissue growth factors that repair the tissues. This is seen in children, but this is not seen, for example, in elderly. So old people, they don't have not either good resistance, not either good tolerance. So this is also something in my lab studies, how virus infects across different ages, and we try to focus in both resistance mechanisms but also tolerance mechanisms. So as I mentioned, now we know what is a virus, now we understand the immune system. So we need to understand the steps of the virus to understand when there's the action from the immune system and recognition and viral clearance. So here are the virus entries. So the first step of a viral infection is the entrance in a cell. So our cells, they have intrinsic barriers. So they have, of course, we have the plasma membrane. We also have this actin cortex, and we also have other intracellular membranes. And the virus use two kinds of mechanisms to enter in a cell. So they can use receptor-mediated endocytosis. So they will bind to a receptor, and this will induce modifications that will allow the virus to generate this endosome and to get inside of the cell. Then we have the second process that is called uncoating. It's when the virus releases the genome already inside of the cell. So they are able to escape from these endosomes, and this could be in a way dependent of pH. So some virus like Zika virus, they really reduce the pH, and they are able to escape from these endosomes, or they can just escape using different kind of strategies. The other way of entering is receptor-mediated signaling. So again, we'll bind to a receptor, and then we'll change the actin, and we'll be able to go directly to the cytoplasm. So here is just to show you like a microscopy. So here is adenovirus. So the adenovirus is here you can see the endosome. So there's a receptor signaling, and then you generate this endosome. And here is herpes virus. And again, the herpes is just entering by fusion with the membrane. And of course, there are viruses that use both strategies, like SARS-CoV-2 use both strategies. And here you can see that there was a release of the genome of the virus, and it's completely empty inside. So this thinking of this is how virus will enter in the cell. So this way we can already have a bunch of receptors on these places that I just mentioned. So I mentioned about the PRRs, the receptors, but these are the pathogen-associated molecular patterns. So these are the patterns that are recognized by the immune system. So this could be glycoproteins that are in the surface of the virus. This could be viral DNA. This could be viral RNA, or this could be other kind of proteins. It just needs to be different from the self. So, and again, where it will be, I know that this is, if you don't work with signaling, this always looks terrible. It's terrible. But you can try just to focus in the way, the molecules that you want. So these are kind of sensors. So as I mentioned before, these are sensors. And here you can see that could recognize different viruses. So we have sensors in the cell membrane, but we also have sensors in the endosome. A lot of viruses use the strategy to go to endosomes, so we'll have a lot of sensors in the endosome. And different from the surface, so usually surface will recognize more glycoproteins that are in the viral structure because it's the outside, the outlayer of the virus that will have contact with the membrane. But in the endosome, the virus, as I mentioned, uncoats, so it releases the DNA or RNA. So the sensors are more, again, the genome. So you have sensors TLR3, 7, 8, 9. They will recognize more, they are DNA or RNA sensoring. And of course, we also have some virus that goes directly. They don't use endosomes, and they go directly to the cytoplasm. And here again, we will have other kind of molecules that can also recognize different kinds of virus. And these will all converge in signaling pathways that will converge in stimulation in the nucleus. I think this will be easier to see here. And this will have a bunch of signals that will probably generate interferon production, cytokines production, that will also then have receptors in other cells, but this will be a signal there is a viral infection going on to neighbor cells. But this is the first line of defense. So here was just to show more like, again, viral proteins will have sensors in the membrane, but we also have sensors in the endosome, probably recognize DNA, probably recognize RNA, and we also have other molecules that are in the cytoplasm, like cGAS, like RIGI, that can recognize also DNA or RNA from virus. MDA5 also recognize, and those also will converge in some molecules that will then produce some cytokines. So again, to understand, oh, I didn't know this was like this. So to understand the next step, we also need to understand the viral replication. As I mentioned, the first step is the attachment and penetration of the virus. And here, what we can also, of course, there are sensing, but antibodies could also prevent virus to attach to the cells. We'll discuss that later, but it's other way. Antibody receptors or antagonists could also block the virus to get into the cells. Then we have the transcription and replication itself. Okay, this will be a little bit slow, but then we have the release of the DNA into the cells. And then we have the translation. And here in the translation, there are multiple innate sensors that can recognize, but we can also produce interferon. There's translation. We have viral proteins that can also be recognized into the cell. Then we also have viral replication, and we have replication inhibitors. Then we have the assembly, when we have the genome and also the proteins being assembled. And then we have the release of the virus. And we can also have, for example, protease inhibitors that will prevent the virus to get out of the cells. Yeah. So, okay. This is already 1 hour and 30 minutes, right? Okay, so we'll finish here. And then we can go back in my next class. But I think this afternoon will be Daniel. But I think you have already some basic concepts, I hope.